well, why you're here?
They're
No, because I am your benefit decided to go on vacation. I'm like, oh, good. So now I can go and talk.
Yeah. And now you're working.
I should have told them I had a problem.
I don't know what's like that.
Okay, are we ready?
Yes.
Maybe. Okay, so tonight what we're starting into is a section on metabolism where we're really getting into some details about metabolism. On the other hand, I'm not going to ask you to draw glycolysis or draw the Krebs cycle. So I want you to know what these things are, and I want you to be able to relate these to cellular respiration and fermentation, hopefully. But don't get too caught up in the chemistry. On the other hand, if you haven't had chemistry for a while, if you have questions about this, don't hesitate to ask. Because we're going through this stuff fairly quickly. And, some of you may need to say, wait, slow down, what are you talking about? Okay. So, we know that organisms that use organic compounds as their source of carbon and energy are nutritionally categorized as chemoheterotrophs. Most of the organisms we work with in this laboratory are chemoheterotrophs. It's also handy because we are. And as a consequence, a lot of biology textbooks, when they start talking about metabolism, they kind of start in the same place. It's like, here, we're all similar. We all do this. I think the biggest problem with it is that we tend to then think that all bacteria are chemoheterotrophs, and they definitely are not. We're looking at a relatively small percentage of them that are. But it's okay. This is where we start. And you can relate it to yourself, which is handy. So bacteria, a lot of bacteria are chemoheterotrophs. Fungi are chemoheterotrophs. Protozoa are chemoheterotrophs. Now, organic compounds have potential energy in them because they're, if you think about carbohydrates or proteins or lipids, a lot of times what you have are carbons bound to other carbons by covalent bonds. When those bonds are broken, energy can be released. So when we say our chemoheterotrophs are getting their energy from organic compounds, that means they're going to take those things apart. And that's what we're going to be looking at. We're going to talk about energy conservation first, the bioenergetics business going on, and then when we kind of finish that up, then we'll talk about the carbon. Because we go back and talk about how do photoautotrophs get their carbon, and then how does carbon come into play with some of these other organisms. So here's a glucose molecule. Actually, that's a model of a glucose molecule. And we do this quite a bit in here, where you'll either see it like this, or you might see just some letters drawn on there. This one doesn't have any letters. Remember, glucose is a hexose monosaccharide, C6H12O6. So there's 6 carbon atoms, and those are going to be taken apart by chemoheterotrophs. This is a really simplistic diagram, and I've showed you this several times, that shows glucose can be catabolized to pyruvic acid. In the process of catabolizing glucose to pyruvic acid, 2 ATP molecules are going to be made. This is a net conservation of energy. So energy that's released from splitting the glucose. But glucose is a 6-carbon compound, and what we get is 2 3-carbon compounds. So we're basically only breaking 1 covalent bond. Covalent bond holding these 2 3-carbon compounds together. When you break that bond, energy is released. This ATP that's made here, this is substrate level phosphorylation. So the energy is coming pretty much directly from the substrate, although as we're going to find out, it's not directly from glucose, because there's a bunch of steps in between. The other thing that happens here is we're going to reduce the coenzyme NAD to NADH plus H+. So remember, coenzymes are enzyme helpers. Their job is to make apoenzymes into holoenzymes. In this case, the NAD is associated with more than one enzyme, and as I said, coenzymes are not nearly as specific as enzymes. So we'll see them in more than one place. This is significant because NADH plus H plus has a higher energy potential than NAD. Which one's oxidized, which one's reduced? You need to remember, this is the oxidized form, this is the reduced form. You look at it, it's like, wait, that molecule looks bigger. Yes, it does look bigger. The reduction doesn't have anything to do with the size of the molecule or the size of the diagram of the molecule. It has to do with that molecule picking up extra electrons. Picks up 2 extra electrons, 1 hydrogen proton. The other proton's floating free, and it's not even shown on there, but usually, well, yeah, it does show 2 floating protons, that's fine. So some of the energy is used to make the ATP. What happens to the rest of it? Entropy. Energy gets away. There's always a certain amount of energy that gets away due to entropy. So they can't capture all of the energy that's released. Now, chemoheterotrophs can make ATP using both substrate level and oxidative phosphorylation. We're going to talk about both types of phosphorylation and take you through the pathways that are involved. So it depends on what kind of metabolism they have. What are the 2 types of metabolism organisms can have? Respiratory or fermentative. So from the last quiz, you might have a clue, but I don't want you to forget that which you learned for exam 1 as we go into the material that's going to be covered on exam 2. Because it's important that you remember the different groups of organic compounds, and it's also important that you remember these categories, the nutritional categories and temperature categories and gas requirement categories, et cetera, et cetera. Don't just let it, go in one ear and fall out the other. Hang on to it. You have to know this stuff for the final. We're going to talk about some of the enzymes that are involved in glucose catabolism, not all of them. And it is significant because glucose is such a commonly used carbohydrate that the enzymes involved in glucose catabolism are for the most part constitutive. What does constitutive mean? Oh gosh, that was just given to you Tuesday. Constitutive enzymes are enzymes that are always being made. The cell needs them. They're essential to cell function, so they're always being made within the cell. They're not something where the production of them can be turned on and off. They're on, basically. Absolutely necessary. So we just answered this already. Organisms can be divided into 2 categories based on metabolism. The types of metabolism are fermentative and respiratory. I think the organisms in the background might be fermentative, that organism in the front, respiratory, but our cells can ferment and do under certain circumstances. Turns out this is true for a lot of organisms. Again, in biology, we like to categorize things, and so we like to say, these organisms are respiratory. Could they possibly ferment? Well, sometimes some of them can. So we are respiratory organisms, but our cells can ferment if they have to. If they're deprived of oxygen, they will make ATP via fermentation. The result is pyruvic acid. It hurts. It causes your muscles to hurt if you exercise extensively and you don't have enough oxygen getting to them. So we're going to start talking about the catabolism of glucose by our chemoheterotrophs, talking about the fermentation type metabolism first, and then we'll talk about cellular respiration. Fermentation is the simpler process. This was defined earlier because we were talking about food microbiology, so I gave you a definition for fermentation. It might not be the same definition that you see if you look up this term in some other reference, but for us, fermentation is defined as the anaerobic decomposition of organic compounds, usually carbohydrates because they're the most abundant organic compounds. And this catabolism involves an organic compound, usually for pyruvic acid, as the final electron acceptor. It's not always pyruvic acid, but it usually is. As far as I know, when we're running fermentation, it is pyruvic acid that serves as the final electron acceptor. The other thing about that electron acceptor, it's made within the cell. So pyruvic acid is made, and then it gets to be used as a final electron acceptor. It's not something coming in from outside. Again, this is showing our very simplistic fermentation pathway. This kind of a pathway might run in an organism like Lactococcus lactis. So what are they doing? They're converting glucose into 2 molecules of pyruvic acid. This is the same pathway I just showed you a moment ago. It's slightly different. If you're not familiar with diagrams like this, pay attention to the arrows. Glucose is being converted to pyruvic acid. The arrow's pointing that way. So one glucose molecule converted to 2 pyruvic acid molecules. In the process, 2 ADP plus inorganic phosphate are converted to 2 ATP. Again, the arrow is pointing down here. And this is a net yield. And 2 NAD are reduced to NADH plus H plus. If it's fermentation, we can't stop at pyruvic acid. We're going to go to our fermentation product, which is lactic acid. And this electron acceptor, the reason this arrow is going back up here, pyruvic acid is our final electron acceptor. What it's going to do is pull the extra electrons and hydrogen, 1 hydrogen proton off of NAD. NAD gets to go back to being its oxidized state, and pyruvic acid becomes lactic acid. So pyruvic acid is a 3-carbon compound, lactic acid is a 3-carbon compound. Lactic acid is the fermentation product in this case. So there we see a diagrammatic representation of pyruvic acid and also a model of pyruvic acid. And then it gets converted to lactic acid. So it's picking up hydrogen and electrons. Huh? Back that up. There's pyruvic acid. Here's lactic acid. Again, I'm not going to ask you to diagram these. And I'm kind of uncertain if it's best to show you these diagrams, because sometimes people then start to freak out about the chemistry. It's like, oh my gosh, do we have to know what these look like? No, not really. Most important thing to recognize is pyruvic acid is a 3-carbon compound. You get 2 3-carbon compounds when you break 1 6-carbon compound. And lactic acid is also a 3-carbon compound. There's not much difference between them. Addition of hydrogen. Now, both fermentative organisms and respiratory organisms are going to use a metabolic pathway called glycolysis. This is a pathway that can be used to catabolize glucose. It's not the only one, but it's the only one we talk about in here because this is an introductory course. What do you suppose that word means, glycolysis? Lysis means to split, and glyco referred to carbohydrate. So literally, glycolysis means the splitting of carbohydrate. Specifically, however, glycolysis is the catabolism of glucose. So it's a metabolic pathway. That means it's going to be a series of chemical reactions that are going to take place. Anytime you have a series of chemical reactions, those chemical reactions require catalysts. In this case, the catalysts are proteins, so they are called protein catalysts. What's the name?
Proteins.
Enzymes. Enzymes. Last time I talked about catalysts, and I said catalysts come in 2 types. If they're proteins, they're enzymes. If they're nucleic acids, they're ribozymes. They're always RNA. And for a while, the RNA molecules were referred to as riboenzymes, and then somebody said, you know what, these really aren't enzymes. Enzymes are proteins. These aren't ribozymes. They're RNA. Anyway, they're not involved in this metabolic pathway anyway. The other thing is, a lot of times in biochemistry, what you'll find out is a metabolic pathway has more than one name. So we call it glycolysis, which is easy. It just means the splitting of carbohydrate. This is also called the Embden-Meyerhof pathway. If you look it up someplace, which we won't, never mind. We don't care. So, glycolysis. This might show up as a definition on a quiz or on an exam. So how would you define it? You can say glycolysis is the catabolism of glucose in 2 molecules of pyruvic acid with the associated production of 2 molecules of ATP, net yield, and the reduction of 2 molecules of NAD into 2 molecules of NADH plus H plus. Because that's what happens, and you can write it out that way, or you could diagram it the way I've diagrammed it several times. It looks like that. If you diagram it, make sure your arrows are going in the right direction. Okay? So what's stated up here is the same thing that's shown here if you're paying attention. One molecule of glucose is catabolized into 2 molecules of pyruvic acid. 2 molecules of ADP plus inorganic phosphate are converted into 2 molecules of ATP. This is a net yield, and 2 coenzymes, NAD, are reduced to NADH plus H plus. It's important to recognize glycolysis stops at pyruvic acid. It can be associated with either fermentation or cellular respiration, but it stops at pyruvic acid. Yeah, pay attention to the direction the arrows are pointing. If you diagram this and you make a line and some curvy things and you don't put arrows on them, you're going to lose points. I want you to know what direction things are going. Okay, so pay attention to the directions on the arrows. Or write it, whichever. As I mentioned earlier, the ATP that is going to be made in association with glycolysis is the result of substrate level phosphorylation. So remember, phosphorylation reactions are those that are going to yield ATP. No, that's not true. Reactions that yield ATP are phosphorylation reactions. Not all phosphorylation reactions yield ATP. You can have a phosphorylation reaction that yields glucose 6-phosphate. We'll get to that in a minute. Anyway, I said there were 3 different types of phosphorylation reactions: substrate level, oxidative, and photo. So substrate level is the simplest, and that's what we're looking at first. Substrate level phosphorylation means the energy that is used to make the ATP comes fairly directly from the substrate being catabolized. It's just that the substrate being catabolized isn't actually glucose. We'll see why in a minute. It is going to be coming directly from a hexose monosaccharide, it's just not glucose. So this diagram is a simplification of the process. It makes it easy, and if you're taking notes, if you write it over and over again, pretty soon you find out it's really easy to write it because you just keep practicing writing it, making sure the arrows are going in the right direction, etc., etc. Yeah, not actually glucose. Again, it's a metabolic pathway, so you can have a variety of enzymes involved. These are protein catalysts. They're made up of amino acids held together by peptide bonds, and most of them are constitutive. The other thing about glycolysis, and this is a kind of a question that I might ask on quizzes or exams, is where does it take place? Well, glycolysis happens in the cytoplasm. Yes?
I'm sorry, in the notes it says although glycolysis is a catabolic pathway, is it both or is it metabolic?
Well, metabolic, remember metabolism is everything.
Yeah.
So metabolism includes both catabolism, breakdown, and anabolism.
Okay.
Right now we're talking about catabolism.
Okay.
However, if you wanted to get into the details of it, when we start looking at individual reactions, some of them are reversible. The overall direction of the pathway, if you are doing glycolysis, going from glucose to pyruvic acid, it's catabolic.
Okay, thank you.
Yeah. So, the enzymes are associated with the cytoplasm in all types of cells as far as I know. Yeah.
And then just real quick, if the pyruvic acid is lactic acid, that'll be a
Right, so then you're going beyond glycolysis.
Okay.
And again, that's really important to remember. Glycolysis stops at pyruvic acid. This little diagram is just reminding you, this is a prokaryotic cell down here. This thing that kind of looks like a koosh ball is the nucleoid. So glycolysis is happening out here. And the prokaryotic cell doesn't have any organelles to worry about. But if that prokaryotic cell happened to be taken up by a eukaryotic cell and it was surrounded by an outer membrane, then that prokaryotic cell would become a mitochondrion. And this becomes significant because, yeah, some of them did a long time ago. Well, anyway. Yeah. So, glycolysis is a catabolic pathway. What we've said about catabolism is oftentimes catabolic reactions are exergonic, so they're going to release energy. But remember, I said that in order for chemical reactions to take place, oftentimes you have to have activation energy. And the activation energy is usually ATP. In this metabolic pathway, ATP is going to be used to phosphorylate first our glucose, and then glucose is going to be converted into fructose, and then that is also phosphorylated. And those steps have to happen before the hexose monosaccharide can be broken down. So there is going to be activation energy involved here. That's why when I said earlier, phosphorylation reactions are those that yield ATP only sometimes. Reactions that yield ATP are phosphorylation reactions, but not all phosphorylation reactions yield ATP. So, in the first part of glycolysis, glucose, our 6-carbon compound, is going to be phosphorylated. The energy of activation is going to be ATP. So ATP is going to be broken, and the phosphate is going to be transferred to ADP. So here again we see this is the first step in glycolysis. Glucose, and I'm not showing you a diagram, but glucose is a 6-carbon compound. It's converted to glucose 6-phosphate. Where did the phosphate come from? It came from ATP, because ATP is being converted to ADP. Again, ATP is adenosine triphosphate, and ADP is adenosine diphosphate. So one phosphate is pulled off, and it's attached to the glucose. Glucose 6-phosphate has a higher energy potential. When we have enzymes that are involved in transferring phosphate groups from organic compounds to organic compounds, sometimes they're called transferases, but I like to call them kinases. And the reason, because we've seen kinesin, we talked about how kinesin was involved in movement. It was moving things along microtubules. So kinase enzymes move phosphates. They move phosphates from one organic compound to another. This is a large group of kinase enzymes. If you get into biochemistry, you find out that each different kinase has a name. I think the first one is called phosphohexokinase. You don't have to worry about that either. Just call it kinase. So kinase is going to move the phosphate. Yep, there it is. It is phosphohexokinase. So it's going to move the enzyme from glucose to glucose 6-phosphate. Ooh, there it is. So that's our first step. We're using some energy of activation, which is ATP. Oh, the other thing is, if you look at that name, glucose 6-phosphate, sometimes people might be confused by that, and they might think the glucose has picked up 6 phosphates. It only picked up 1. The 6 in the name tells you where that phosphate was attached. So it's attached to the number 6 carbon. So glucose picks up 1 phosphate, it just happens to be attached to the number 6 carbon of the glucose. So there you can see it attached. So if you count on the glucose molecule, the carbons are not shown here, but at each angle there's a carbon. So this is 1, 2, 3, 4, 5, and here's number 6. Now that's where the phosphate is. So glucose 6-phosphate. The next thing that's going to happen is the glucose is going to be converted into fructose. Let's see what happens next. Well, anyway, that's our energy of activation. So next, glucose is converted into fructose. Fructose and glucose are isomers, which means they both have the same molecular formula, C6H12O6. The difference is the position of hydroxyl groups. When a molecule is converted into its chemical isomer, it doesn't require energy, but it does require an enzyme. And the enzyme in this case is called an isomerase. So again, remember enzyme names end in ASE, and they tell you what the enzyme does. So an isomerase enzyme is an enzyme that's going to convert a molecule into its chemical isomer, in this case glucose into fructose. But it was glucose 6-phosphate. When it gets changed, it's changed into fructose 6-phosphate. Again, don't worry about the diagrammatic representations of molecules. I'm not going to ask you guys to draw this. Furthermore, I'm not going to ask you to draw the glycolysis pathway. I just want you to understand that it is a multi-step pathway. It happens in the cytoplasm, and you need to know a couple of enzymes that are involved. The enzymes you need to know are kinase enzymes and isomerase enzymes, and they're used in more than one place. So again, there's no energy involved in this. The cell didn't have to use energy to convert glucose to fructose. It also didn't get any energy from doing that. There's no energy involvement. And furthermore, back the truck up a second. If you look at that pathway, technically that arrow could go either way. That would be a reaction where you would have an equilibrium. You can either go to glucose 6-phosphate or fructose 6-phosphate. Either direction, that isomerase can go either way depending upon concentration. The next thing that's going to happen is a second phosphate group is going to be added to the fructose. So the fructose is going to be going from being fructose 6-phosphate into fructose 1,6-bisphosphate. In older textbooks, it used to be called diphosphate, which made sense to me because you think di means two. I don't know why it's this. They changed the name, but it is now bis. Anyway, because we're going to be transferring another phosphate from ATP, converting fructose 6-phosphate into fructose 1,6-bisphosphate, again, ATP is going to be converted to ADP. Since we're taking the phosphate off of here and sticking it on here, that is a transfer of a phosphate group from organic compound to organic compound, and the enzyme involved is a kinase. It's a different specific kinase, but again, for you guys, that doesn't matter. It keeps showing it on the slide though. Oh yeah, that's phosphofructose kinase. Really, you don't need to know that. Just know that it's a kinase. That's fine. So now fructose has a phosphate at each end. Again, the 1,6 tells you where the phosphate is attached. There's one phosphate on the number 1 carbon and another phosphate on the number 6 carbon. And we've used up 2 molecules of ATP to get to that point. So all we've done is add activation energy to this system. From here on, we can start taking things apart. So our 6-carbon compound, fructose 1,6-bisphosphate, is going to be split. And it actually gets split into 2 molecules that are not the same. So it's going to be split into 2 molecules. One is called glyceraldehyde 3-phosphate, and the other is called dihydroxyacetone phosphate. And there is an arrow here showing you that these 2 molecules are in equilibrium. The other thing I should point out is that glyceraldehyde 3-phosphate has another name. It's called phosphoglyceraldehyde, or PGAL. Different biology textbooks use different acronyms for that molecule. You'll see different ones depending on which book you look at. That's significant to mention because there's actually a glycolysis song, and at a certain point they start talking about GLP. GLP is glyceraldehyde 3-phosphate. If they talk about PGAL, PGAL is phosphoglyceraldehyde. It's the same thing. Anyway, well, so then typically what will happen is you'll see diagrammatic representations where you're just following this side of the pathway. So fructose 1,6-bisphosphate is actually divided into these 2 molecules. This one is the one that's going to be used to go down the rest of the pathway. But as soon as you remove some of this becomes this because they're in equilibrium. These are isomers. Consequently, what is that enzyme? It's an isomerase, right? You don't need to know that one either, other than it's an isomerase. So there it just says glyceraldehyde 3-phosphate is also called phosphoglyceraldehyde. Now, in the lecture syllabus, you can see diagrammatic representations of glycolysis, and I don't think the diagram that's in there has all the steps. It's kind of a simplified version of this, which is not a big deal. Again, this is not a biochemistry class, so you're not expected to know every single enzymatic reaction. Neither are you expected to know every single enzyme. You are expected to know we have to have kinase enzymes for glycolysis, and we have to have isomerase enzymes. Again, that's just telling you that those 2 molecules are in equilibrium, and that the enzyme that's converting one to the other is another isomerase. Yes?
Sorry, isomerase, all we need to know about that is that
Just know isomerase.
that's all we need to know. Okay.
Yeah. Don't worry about the specific isomerase.
Just that it's an isomerase.
Right. Part of the reason I don't want you to spend a lot of time looking at that is because depending on which reference you look at, enzymes can have different names. Usually, well, like I said, a kinase can also be called a transferase. We're gonna call them kinases,
Okay.
but different texts will give you slightly different versions of the specific name. And again, it's an introductory class. As long as you know kinase, a kinase is gonna transfer a phosphate group from an organic compound to an organic compound.
Thank you.
So, the enzyme that's involved with that breaks down the fructose to two different molecules is isomerase.
No, the isomerase is here, between these two. I didn't give you the name of the enzyme that breaks the fructose.
Okay.
So don't worry about it. There's gonna be a lot of enzymes in here that you don't need to know.
Okay.
Again, because this isn't biochemistry. And you have to know enough. Okay. Now, the next thing that's gonna happen is these molecules of glyceraldehyde 3-phosphate, or phosphoglyceraldehyde, depending on which you want to call it, they're gonna be phosphorylated and oxidized, and they're gonna form two molecules of bisphosphoglyceric acid, or PGA. This is the process where the coenzyme comes into play. Again, the enzyme involved here, you don't need to worry about the name, but you do need to notice what's going on here, because NAD is picking up electrons and hydrogen protons, and it's becoming NADH plus H plus. This is significant because this is a higher energy form molecule, the reduced form of the coenzyme. So some of the energy that was released is now captured as reduced coenzyme. That is energy that was released when we took our 6-carbon compound apart. It doesn't happen immediately, but it happens. Nevertheless, that's not phosphorylation, that's reduction. The other thing that's significant here, that you don't have a kinase involved in the reaction of making a phosphorylated molecule this time around, because the energy is coming from inorganic phosphate, inorganic pyrophosphate. That's not something I'm ever going to ask you on an exam, so don't worry about it. The only time that would come into play is if you started looking at the pathway and thinking, huh, how did that happen? Where did phosphate come from? It comes from inorganic phosphate, pyrophosphate. And then, remember we talked about phosphate being stored within metachromatic granules, within prokaryotic cells? So presumably they have excess stores of phosphate that they can use to make this. But again, don't worry about it. These things were also used to make our high energy compounds, the nucleotide triphosphates, ATP, GTP, CTP, etc. Pyrophosphates are the last two phosphates added. So they exist naturally. Now, another kinase is going to be involved in the next reaction. So again, a kinase is going to be transferring a phosphate group. In this case, we're going to be transferring a phosphate to ADP to make ATP. So our bisphosphoglyceric acid is going to be converted to 3-phosphoglyceric acid. Again, when you see bis in front of the word, it means it had 2 phosphates. So that glyceric acid had 2 phosphates on it. It goes to phosphoglyceric acid. Now there's only 1 phosphate on it. Where did the other phosphate go? It got onto ADP to make ATP. So if we're transferring the phosphate from here to here, converting this to ATP, the enzyme involved is a kinase. And again, don't worry about which kinase, just know that it's a kinase. The other thing to remember, because each glucose molecule that is metabolized yields 2 P-gals or 2 G3Ps, you have to follow each one down the glycolysis pathway. And so we're going to be phosphorylating. We're going to be making 2 molecules of ATP at this point, phosphorylating 2 ADPs. This is showing the same diagrammatic reaction. Notice that we're converting ADP to ATP. Where did the phosphate come from? It came from the bisphosphoglyceric acid. Am I ever gonna ask you that on an exam? No. We're just going through the steps. So you see that this is a lengthy pathway, perhaps so that later when you're studying, you can go, I'm really glad we only have to know glucose, arrow, pyruvate. Like not all the steps in between. Yeah. Okay. The next thing that's going to happen is 3-phosphoglyceric acid is going to be converted to 2-phosphoglyceric acid. This involves a mutase enzyme. You don't need to know that one either. And then we're going to form phosphoenopyruvate, PEP. Yeah, do you have to know these enzymes? No, you don't. Nope. Nope. You only have to know 2. This is not a biochemistry class. However, do you remember me mentioning phosphoenopyruvate ever before? Probably not. It was one of those little things that kind of slipped by when I was talking about transport mechanisms. And I said that bacteria can do a transport mechanism called group translocation, where they take phosphate from phosphoenopyruvate, PEP, and they stick it on glucose. So they can bring glucose into the cell and convert it to something else, which allows them to constantly maintain a low level of glucose inside, because what's inside is immediately glucose 6-phosphate. Trivia. Microbial trivia. Okay, so then the last step, we want to get to pyruvic acid, which is our end point on the glycolysis pathway. So the last step is going to involve pulling a phosphate off of phosphoenopyruvate and transferring it to ADP. So 2 phosphoenopyruvate molecules get converted to 2 pyruvate molecules. Again, the phosphate that's coming off of this organic compound, that phosphate is going to get stuck on ADP, converting it to 2 ATP. So what's the enzyme involved? Kinase. The kinase enzymes are very significant then. Kinase enzymes are involved in transferring phosphates. And this is the end point, because we converted glucose to 2 molecules of pyruvic acid, so we're done. That's glycolysis. That's handy. So in that last step, we got to make ATP, again because you get 2 molecules from a single 6-carbon compound, you end up with 2 molecules of ATP, or excuse me, 2 molecules of pyruvate at the end. We actually made 4 molecules of ATP total. We made 2 when we did the first conversion of ADP to ATP, and we make 2 more with this second conversion of ADP to ATP. We made 4 ATP, but it took 2 ATP to get this process going. We had to phosphorylate glucose, and then we had to phosphorylate fructose. So when I say that the net yield of ATP for this process is 2, it's because you have to subtract 2 from 4. There's the glycolysis pathway. Do you have to draw it? No. That's the good news. I keep showing it to you. I just want you to appreciate it and the fact that this is a microbiology introductory class, not biochemistry. So you don't have to memorize all those molecules, you don't have to memorize all those enzymes, but you do have to know, where does this happen?
Cytoplasm.
Cytoplasm. All kinds of cells, prokaryotic cells, eukaryotic cells. Well, as soon as I said it, it's like, wait, not all kinds of cells, because not all kinds of cells are chemoheterotrophs. But if we're talking typical chemoheterotroph, they're going to have the enzymes to run the glycolysis pathway, and the glycolysis pathway enzymes are in the cytoplasm. They're not in some compartment someplace, they're just in the cytoplasm. And we know that we're going to start with glucose, a 6-carbon compound. We're going to end up with 2 molecules of pyruvate. These are 3-carbon compounds. Basically, we broke 1 covalent bond, but we did it in a whole series of steps. In order to run this reaction, we had to have activation energy. The activation energy was supplied by ATP. And we're going to get 4 ATP out, but we had to put 2 ATP in. So our net yield is going to be 2 ATP. The other thing that is significant about this pathway is we're going to reduce the coenzyme NAD to NADH plus H plus, and that is potential energy. So we've got that stored up. And we do that twice. We got 2 NADH plus H plus produced because Glucose is a 6-carbon compound, and when you take it apart, you get 2 3-carbon compounds. Once you get to the 3-carbon compound part of the pathway, you run it twice. In other words, you have to follow 2 lines down from glyceraldehyde 3-phosphate or phosphoglyceraldehyde. It doesn't matter. DHAP is going to be converted to PGAL or glyceraldehyde 3-phosphate because that's an isomerase running that reaction. Is there anything else you need to know about this? Oh, yeah, there is. There's a rather major thing that you need to know about this. So the enzymes you need to know, kinase and isomerase. Not the specifics on those, just kinase and isomerase, so you're good. But this pathway cannot run by itself. In other words, it cannot run indefinitely without being connected to something else. And this is very significant. We said that living organisms have to have either a respiratory or a fermentative type metabolism. This is the first stop or the first step in both fermentation and cellular respiration. If you're talking about the catabolism of glucose, oh, back to what you need to know, yeah? Can you define it? You gotta know where it occurs. Gotta know 2 types of enzymes. Here we are. Can this pathway continue without additional chemical reactions taking place? The answer is no, it cannot. Any guesses why? It's a catabolic reaction. It does take ATP as activation energy, but you get more out than you put in, so that's not the deciding factor. But remember the spoons in the peanut butter? You come into your kitchen and all your spoons have got peanut butter already, and you wanted to use that spoon to measure something else. You've got to get the peanut butter off of there. That was a terrible analogy. So, in this view of the pathway, we went down, we took glucose apart, we get our 2 3-carbon compounds, and then we're following this down. Notice over here, we went to 3PG there, and both on this side too. We're reducing NAD here, we're reducing NAD here. This reaction to go from here to here requires that coenzyme, and it requires that coenzyme in the oxidized state. In other words, it requires a clean spoon. You've got to get the peanut butter on. You have to have an electron acceptor.
Can you go back 1 slide so I can like see that question, how you asked it? Please.
Okay, let's go back and talk about what you need to know, so you don't freak out about glycolysis pathway. Quick, you need to know, what is it? In other words, define it. You can either write out the description or draw the picture. Glucose. 1 glucose goes to 2-pyruvic acid, conversion of 2 ADP to 2 ATP, conversion of 2 NAD to NADH plus H plus. With the arrows pointing in the right direction. Where does it occur? In the cytoplasm. All kinds of cells that run this pathway. 2 types of enzymes. Kinase, transfer phosphate groups between organic compounds. Isomerase, convert molecules into their chemical isomers. And then can it run without being connected to something else? And the answer is no. It cannot, because the coenzymes, which are required for that step that was shown in there, those coenzymes have to be available in their oxidized state. You have to have NAD, not NADH plus H plus. So this pathway, was that long enough?
Yes, thank you.
Okay. This pathway, this glycolysis pathway, starting with glucose at the top and going down here to 2 pyruvates at the bottom. Again, we make ATP here, ATP here, ATP here, ATP here. We make plenty of ATP to cover our energy needs because it only took 2 to get the thing going. Activation energy, 2 molecules of ATP, we get 4 out. The stopping factor is this: NAD reduced, NAD reduced. We need a final electron acceptor. The final electron acceptor is different if you're running a fermentation pathway than if you're running a respiratory pathway. If the organisms are fermenting, their final electron acceptor is organic. And it's inside the cell, in this case because they made it. What's the final electron acceptor if we're fermenting? The final electron acceptor. On the picture. It's like,
It's NAD, right?
no.
What is it?
NAD is the coenzyme.
Okay,
The coenzyme got reduced by picking up extra electrons.
okay.
Became NADH plus H plus. What's confusing there, I think, is you get to see the hydrogen stuck on there. You can't see the electrons. Believe me, they're there. It picks up 2 electrons, 1 hydrogen. That's why that other hydrogen is floating out there, that proton. So, our final electron acceptor is, somebody over here said it. Yeah, say it louder. Pyruvate. Final electron acceptor is pyruvate. Pyruvic acid, pyruvate is just the salt of the acid. Don't worry about interchanging those words. That's fine. So, pyruvic acid or pyruvate is going to be our final electron acceptor, and it's going to allow our coenzyme to be reoxidized, which is essential because without oxidizing the coenzyme, glycolysis is going to stop. Cell only has so much coenzyme.
How does it get more? If glycolysis keeps going and going and going.
Well, they can make it. Remember, it's derived from vitamins.
But they won't make it as they need it to
They
keep the
can't make it as fast as they need it to run metabolism, because metabolism is happening really quickly. So they need a way to get the extra electrons off of that thing. So basically, they're recycling their coenzyme. Eventually, it will break down. Besides, remember, these cells can undergo fission every 20 minutes. We're talking bacteria. They have to make more. But they make it from B complex vitamins?
And they don't make it fast enough to make the glycolysis cycle.
Apparently not. No. So they're very efficient. So they're gonna want to recycle that coenzyme. And the easiest way to do it is to take the electrons off and transfer them to something else. So there we see the pathway again. Just a summary, it's going to yield 2 ATP, 2 pyruvate, 2 waters, 2 NADH plus H plus. Is this a problem? Only in the sense that your coenzyme has to be in the oxidized form to run that pathway. So we have to get it back to the oxidized state. Otherwise, glycolysis is going to stop. So the most important thing I'm trying to get across here is glycolysis can't stand alone. This is significant because we said we've got 2 different types of metabolism. Fermentation and respiration, or oxidation, depending on what you want to call it. Cellular respiration or an oxidative metabolism. If the organisms are fermenting, then they're going to let pyruvate be the final electron acceptor, and they're going to produce something else. But again, glycolysis can't stand alone. Why not? You have to have a way to oxidize the coenzyme. It's the coenzyme that is the limiting factor here. So you have to have a final electron acceptor to remove the electrons and hydrogen protons from NADH plus H plus. Put it back to the oxidized state, NAD. And here we see that happening. Again, our final electron acceptor is pyruvic acid. Handy, the cell just made it. And it can use the electrons. It can take the extra electrons and hydrogen from here, and it becomes lactic acid, which is a fermentation product. So at that point, we have made our pathway. We started with the glycolysis pathway, and now we have converted this into the fermentation pathway, just by adding that last step. But what's important is to think, okay, what was fermentation? It's the catabolism of organic compounds that involves an organic compound, usually pyruvate, as the final electron acceptor. And the electron acceptor is required to get NADH plus H plus back to NAD, so that the glycolysis pathway can continue. Am I getting through to you guys? A lot of puzzlement there. A lot of people going, huh? This is not easy. Believe me, it's not easy, especially the first time you run into it. So I'm not sure if this is still pounded into people in every class. It used to be part of physiology. I don't think it was part of anatomy, but is it still in physio?
Yes.
Yeah.
Good, because it needs to be. And I present it perhaps a little bit differently simply because I'm focusing on microbes, but our cells can do that too. Our cells catabolize glucose to make ATP, and if they don't have a good supply of oxygen, they will ferment, and they will use pyruvate as a final electron acceptor, and you will get lactic acid as a byproduct. Now, the term that I gave you, and this I did mention in lab when we were talking about food microbiology, if organisms produce lactic acid as their only fermentation product, then we say they are homofermented. Remember, homo means same. It means the only metabolic pathway they have is going to be this one. They're going to produce lactic acid as their only end product. So homofermentative organisms might look something like that. The ones that we've talked about thus far included Lactococcus lactis. Remember Lactococcus lactis and Lactococcus lactis, lactis lactis and Lactococcus lactis cremoris. They're the primary fermentative organisms associated with cheese production, and lactic acid is one of the primary flavor components of cheese. So salt and milk type can influence cheese flavor, but a lot of the flavor is lactic acid. So it's that sour flavor that we kind of like. The Leuconostoc mesenteroides and Lactobacillus brevis and plantarum, what are they associated with? I heard it whisper.
Cheese.
No.
Sauerkraut.
No. Sauerkraut. Sauerkraut. So they came free with the cabbage. It was a big deal. You chopped up the cabbage and you didn't have to worry about putting organism with it because they came with the cabbage. They came free. Good deal. You get some cabbage, you get some free bacteria. Of course, you know, those things are everywhere. And they also produce lactic acid. How many of you actually got to look at those organisms last time? So that lab was chaotic, and I apologize. So I looked at the clock and just went, what? It's 10:30? How'd it get to be like that? I'll try not to do that to you again tonight. Sorry, that's very bad. Well, anyway, if when you get to look at these things, your cheese organisms and your yogurt organisms. So the cheese organisms are supposed to be Lactococcus lactis. Lactococcus was initially categorized as a streptococcus. They do form gram-positive cocci in chains. So they would look kind of like this. That's how you, kind of. Except they're all going to be on the same plane. This is an artistic representation, you know, where you're looking at the edge of this cell. This guy's really close to you. And then these are not quite so far away, and then these are further and further back. People have such fun with bacteria. Yours are all on the same plane because they're stuck to a piece of glass. Anyway, gram-positive cocci in chains, Lactococcus lactis. The Lactobacillus and I think Leuconostoc are also cocci, but the Lactobacillus are bacilli, so they're going to look more like this. Then again, the ones that we find in association with yogurt usually don't look like these little short rods. They look like purple spaghetti. They're big, long rods, little skinny guys. They're kind of fun to look at. Anyway, they're mostly homofermentative. Now, this is an alternative pathway. If organisms produce fermentation products other than lactic acid, we say they're heterofermentative. Hetero means different. So in this case, we see glucose being converted to pyruvic acid, and then pyruvic acid being converted to acetaldehyde. Acetaldehyde is a 2-carbon compound, so one of the carbons came off, and it went that way as CO2. Now acetaldehyde is going to be our final electron acceptor. It's going to take the electrons from NADH plus H+, allowing that coenzyme to be oxidized, and acetaldehyde is going to be converted to ethanol. Who does that? Saccharomyces cerevisiae. We didn't get to sample our wonderful food products last time. I'll try to get the cart we have the table cloth back. I'll try to get that set up in case anybody's dying to sample sauerkraut and poor quality apple wine. Last semester somebody said, it tastes like wine or the stuff that comes in a box. Anyway, here's Saccharomyces cerevisiae doing its fermentation thing. So at the top you see Saccharomyces cerevisiae being used to produce beer. The bubbles are CO2 and ethanol is staying in the liquid. Same thing happening in our wine flasks. And then on the bottom picture, we see bread that has risen up making the fluffy bread, that's again CO2. So it makes the dough rise. In that case, when you bake it, the ethanol all goes away. But we like their products. Some of us like them more than others. Now, this is kind of an interesting diagram because this shows you some of the possibilities for organisms that are heterofermentative. What's important to know here is that this is the kind of thing that you can expect from our fermentative organisms that we use for physiological unknown 1. Everybody has one organism type that is gram-negative and fermentative, and they are not homofermentative. They are heterofermentative. So later on, we're going to be doing some enzymatic tests where you're finding out, can these organisms produce acids? And the acids that they produce, you can't necessarily assume it's lactic acid, because that's not likely to be what they produce. They could, so they can take glucose to pyruvic acid. From there, they can go to succinic acid, which can be converted to propionic acid. They can convert pyruvic acid to acetylmethylcarbinol, which is also called acetylene. They can convert pyruvic acid into acetyl coenzyme A, form acetoacetic acid or acetic acid. They can form lactic acid, but they can also form formic acid, acetaldehyde, ethyl alcohol, isopropanol, acetone, butyric acid, butyl alcohol, or beta-hydroxybutyric acid. There's CO2 being produced here, up here, hydrogen. So we will be testing to see if these organisms can produce gas. If they can produce gas, we just have to say, yeah, they make gas, because we don't know if it's CO2 or hydrogen, and we're not going to be doing any kind of test to determine. So our heterofermentative organisms, when they make acid, you can't say it's lactic acid. It might be, but it's probably not. And this byproduct over here, acetylmethylcarbinol, this is a specific test that we'll be doing. It's called the Vokes-Proskauer test. We're testing for acetylmethylcarbinol. Not all organisms can do that. So we'll get to do a variety of different tests to find out what these things can do, and that's how you will identify them. Yeah, that's what that is. I just looked. Okay. So the advantages to fermentation. It allows organisms to make ATP, so they're going to be catabolizing organic compounds. They are taking the organic compounds apart so that they can capture the energy, the ATP. They need ATP to drive anabolic reactions, to move their flagellum, to do what active transport, whatever. However, fermentation is not very energy efficient. We took a 6-carbon compound and we split it into 2 3-carbon compounds. If we ferment, we don't necessarily stop at pyruvic acid. In fact, we can't. But even if you go to lactic acid is still a 3-carbon compound. Only one covalent bond was broken. If they pull off a CO2 and make acetaldehyde, they don't get to capture any more ATP for doing that. They're not going to be able to use that reaction to capture any more energy. Consequently, the energy efficiency of fermentation is pretty low. It's advantageous because the organisms can do it under anaerobic conditions. They don't have to have oxygen available for them to do this. But again, not terribly energy efficient. The other disadvantage of fermentation is that the cells are going to be potentially making some pretty nasty byproducts. So last time we talked about organisms in the stationary phase, if they're in a batch culture, where they will stop growing due to lack of nutrients and the buildup of toxic metabolic waste. What are some of those toxic wastes? Organic acids. That's going to drop the pH, and pH is one of the factors that influences enzyme activity. If the pH becomes too acidic, they can't work anymore. The other kind of fermentation product could be ethanol. Ethanol is toxic. It's toxic to us too. If you drink too much of it, we call it intoxication. It'll kill you if you do it too often, too long. Sustained consumption that destroys your liver. It's not good for you. But anyway, so there are these potential toxic wastes. If the bacteria are living in a natural environment, let's say they're out on your lawn under an apple tree, and an apple falls down there and they're doing their catabolic thing, probably the waste products are going to be diffusing away into the soil, and it's not going to bother them that much. It bothers them a lot when they're stuck in a beaker. So our organisms that are back there playing with the cabbage, they will reach a point where the acid they've produced in there will kill them, and then they stop. But then the cabbage is preserved. Same thing's happening to our apple juice. Our apple juice is being converted into apple wine, but the organisms are producing ethanol, and it will eventually kill them. I'm hoping they haven't died yet, because the wine tastes better if it doesn't taste like dead yeast. Still tastes like rubber stuff. That's okay, we don't want you guys drinking too much of it. Okay, so that's fermentation. The alternate form of metabolism is respiration. So if you are studying physiology, there's 3 levels of respiration, right? External, internal, cellular. So when we talk respiration in here, we're talking cellular respiration, most of the time because the organisms we're dealing with are single-celled organisms. But for those of you who are not familiar, external respiration is getting oxygen from the outside environment into your lung and then into your bloodstream. Internal respiration is getting it from the bloodstream into your cells. Cellular respiration is what happens in the cells, and that's what we're going to talk about. For bacteria, oxygen comes in how? What's the process? Oh my word. Gases, oxygen and CO2 get into cells by means of diffusion. Simple diffusion. They simply diffuse down their concentration. Don't forget the stuff you studied and learn for exam 1. So now we're going to be talking about cellular respiration. This is happening inside the cells. And again, it's a multi-step process. What's really nice about cellular respiration is it also involves glycolysis, and we've already talked about that. Woo-hoo. So we've covered 1/3 of it. Cellular respiration is going to allow organisms to completely catabolize glucose, and this is a big deal. Glucose is a 6-carbon compound. When we fermented, we simply tore it apart. We made 2 3-carbon compounds. We only got 2 molecules of ATP. Now we're going to take apart glucose 1 carbon at a time. Every covalent bond that's broken is going to release energy that the cell is going to get to capture. Well, they don't get to capture all of it because some gets away due to entropy. But they're basically going to convert glucose into carbon dioxide. So here's our diagrammatic representation. Glucose is converted to carbon dioxide. Actually, this is a pretty significant diagram because it tells you where carbon dioxide is coming from. Most of the carbon dioxide that is released by living organisms is coming from the catabolism of organic compounds. We're going to see glucose is not the only thing that can be catabolized, it's just the most abundant. But this is important because some people think that you breathe in oxygen, O2, stick a carbon on it and breathe it out. You don't. And if you have that in your brain, take some kind of sharp implement and dig it out and throw it as far as you can. Because if you put that on an exam, I'm going to write no, no, no with exclamation points behind it. We do not. Okay. So glucose is going to be catabolized. ADP plus phosphate are going to be used to make ATP, and we're going to make a bunch of ATP, either 36 or 38, depending on what cell type we're looking at. Those are, by the way, those are maximums, and the 38 is accomplished by prokaryotic cells, not eukaryotic cells. I'm going to explain why in a little bit here. But that's a heck of a lot more than 2. It's still not 100% efficient. It's close to 40% efficient, only because 38 is close to 40. But you're not going to have 100% efficiency because entropy happens, some energy gets away. Okay. Now, we took about glucose and we converted it to CO2. If molecular oxygen is involved as the final electron acceptor, then water is produced. But if it isn't, water is not produced. So you have to be careful about summary type diagrams. So some references show you cellular respiration looking like this. This is from a general biology textbook. And what it says is you take glucose, C6H12O6, and you bind it with oxygen, 6O2, and you will make 6 CO2 and 6 waters, and you're gonna release energy. It's handy, but it's not necessarily accurate. It depends on what the organisms are using as their final electron acceptor. We use oxygen. What do we do with the oxygen? I just told you where CO2 comes from. So what do we do with the oxygen? How good's your chemistry balancing abilities? Sugar, 6 C's go to here. 6 CO2. And the O2 is associated with the carbon as a carbox group, COOH. Consequently, these 6 oxygens are going to be converted into what?
Water.
Water. Exactly. More chemistry than we can stand. Ah. Anyway, they only do that if they're using oxygen as a final electron acceptor, and some bacteria can use nitrate. That's another test we get to do in here to find out, can they use nitrate? The significance of this, what comes to mind immediately is Pseudomonas. Pseudomonas is an organism that is described as being obligately aerobic. That's not true. Pseudomonas are respiratory, but Pseudomonas in a clinical setting can sometimes be found very deep in tissue, living in the same environment that supports Clostridium. And Clostridium are obligately anaerobic. And then people find it in there, and it's like, wait, how can this be? Pseudomonas are supposed to be strictly respiratory. They are, but they can use nitrate as a final electron acceptor. They don't have to have oxygen. Another piece of microbiological trivia. Anyway, so again, that diagram is accurate only if the organisms are using oxygen as a final electron acceptor. We do, but not every organism does. Again, this class we're going off on, and yet you always have to remind yourself, think outside your own personal being, because microbes do some pretty weird things. So, cellular respiration, it's a multi-part metabolic pathway. The 3 parts are glycolysis, the Krebs cycle, and the electron transport chain, which is also called the respiratory chain. I wish it had a better name, but as far as I know, it doesn't. So 3 parts, glycolysis, Krebs cycle, electron transport chain. Again, the good news is we already looked at glycolysis, so we know what happens there. Just for your review purposes, glycolysis is the conversion of 1 molecule of glucose into 2 molecules of pyruvic acid with the associated production of 2 ATP molecules, net, and the reduction of 2 NAD to NADH plus H plus. Sure your arrow is pointing the right way. You can just draw it like that. Now, what we're interested in finding out is what happens to the pyruvate. Yeah, if you want to draw it like that, sure, go for it. Probably not. I remember taking a class where it was like, really? We're supposed to memorize that? Yes, that's what biochemistry does for you.
You were taking it or teaching?
I was taking it, biochemistry. That was one of those nightmare type classes, you know. Oh gosh. Anyway, so the next step, the Krebs cycle. It's also called the citric acid cycle. It's also called the tricarboxylic acid cycle. We have a board in here that has it on it. But unfortunately, a lot of our coenzymes have been removed from the board. So here you can see the Krebs cycle. The reason it's called tricarboxylic acid cycle is because the first acid in the cycle, which is citric acid, has 3 carboxyl groups. So tricarboxylic. Krebs cycle because apparently it was a Dr. Krebs that figured out most of the cycle. Citric acid is the first acid, so it's also called the citric acid cycle. Darn those chemists. They have all these different names for things. Okay. So that's just what I just said. citric acid cycle, tricarboxylic acid. Krebs cycle is easier to spell. Anyway, most of the energy that is released from the catabolism of organic acids during this pathway, most of the energy is going to be captured in the form of reduced coenzyme. Some biology textbooks try to tell you that the Krebs cycle releases energy and it's mostly captured in the form of ATP. No, it isn't. Very little is captured in the form of ATP. Most of it's captured in the form of reduced coenzymes. Remember that I said these reduced coenzymes, NADH plus H plus and FADH2, those have energy potential. That's going to be significant because when we get to the last step in cellular respiration, which is the electron transport chain, those coenzymes are going to be able to transfer energy to a series of electron acceptors, and you're going to end up making ATP via oxidative phosphorylation later. Okay, here's the Krebs cycle, citric acid cycle. Again, not a pathway you need to draw, but it's a good idea to recognize that it is cyclic. Well, that's easy, the name says it is. This thing starts with a 6-carbon acid over here, citric acid, and what happens around this pathway is we pull carbons off. Pull off a carbon here, pull off a carbon here. The initial carbon that got pulled off is pulled off up there. So, if you take glucose apart and you get 2 molecules of pyruvic acid is a 3-carbon compound, and you pull off carboxyl, 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 you've completely catabolized that thing. And that's what's going to happen. Now, between glycolysis and the Krebs cycle, there's an important intermediate step. And this intermediate step is different depending on whether the organisms are running fermentation or running cellular respiration. Since we're talking cellular respiration, we're going to be talking about this step, where we're going from pyruvic acid to acetyl coenzyme A. It's kind of hard to follow the arrow, but what we're doing is going from here to pyruvic acid to here, to acetyl coenzyme A. Along the way, we have to add a coenzyme, coenzyme A, and we have to remove a carboxyl group, which is going to come off over here as carbon dioxide. And we're also going to reduce NAD to NADH plus H plus. I believe in the lecture notes there's an error that tells you the enzyme that is running this particular set of reactions. It's a complex, it's an enzyme complex, so it's more than one step. But I believe your lecture notes say that the enzyme involved is pyruvate decarboxylase, and that's wrong. The enzyme that is responsible for running this pathway is called pyruvate dehydrogenase. Pyruvate decarboxylase is the one that pulls the CO2 off for fermentation. You get CO2 and acetaldehyde. So it's a different enzyme. I apologize. So this is the correct enzyme complex, pyruvate dehydrogenase. Watch out for the lecture notes.
They said in place of decarboxylase.
Yes, pyruvate decarboxylase is the one that's involved in the fermentation pathway. You're going to probably run into this not only in the lecture notes, but also in the study guide. It's one of those places where I know there's an error. It's like, why don't you get in there and fix it? Well, I should. I'm trying to tell you guys, watch out for it.
What's the other enzyme that's not part of? You were correcting what enzyme again?
So the enzyme that would have pulled off a carbon dioxide and converted pyruvic acid into acetaldehyde, that's pyruvate decarboxylase. Pyruvate decarboxylase doesn't do the reaction that you see here, where you convert pyruvic acid into acetyl coenzyme. And I'm pretty sure old textbooks said it did, but it doesn't. So this is something that has been sorted out since, I guess. You can see this kind of reaction. I think you can actually Google and find little short films that show this happening. So it's more than one step. But again, pyruvate is a 3-carbon compound. We're gonna pull off 1 carbon and it comes off as a carboxyl group, COOH. The extra electrons that are on that and 1 hydrogen proton get passed to NAD so that NAD gets reduced. But it is significant that the carbon comes off as a carboxyl group. It doesn't come off as just carbon alone. So that first CO2 that's produced up here, where did the oxygen come from that is associated with the carbon? It came with it. It came with it off the acid. And we add a coenzyme A, to make acetyl coenzyme A. An acetyl group is a 2-carbon group. So we had a 3-carbon pyruvic acid. We pull off 1 carbon as carbon dioxide. We have a 2-carbon left, and that 2-carbon binds with coenzyme A to form acetyl coenzyme A. Again, you need to know this enzyme, pyruvate dehydrogenase. It's pyruvate dehydrogenase complex. There's a homework assignment coming up. It's in the lecture syllabus. It's a homework assignment that has to do with glycolysis and cellular respiration. We're not going to finish this tonight, but that's okay. Yep, so here it is telling you, the lecture notes tell you. See, I fixed it on here. Why didn't I fix it in the lecture notes? I don't know. This is easier, I guess. My mistake. It's not been corrected. You guys corrected.
Wait. Dang it, it's not working.
I'll catch that slide.
Slow. I got it. Okay. Alright.
That's fine. That's fine, I'm trying to think. Okay, so the quiz that you're gonna have on Tuesday. This is a very important enzyme complex. So don't forget, it's not pyruvate decarboxylase. It's pyruvate dehydrogenase.
Are we gonna be
Are you gonna be quizzed on this? Sure you are.
no on decarboxylase?
No,
Okay.
because
I don't know if they're
no.
two separate things or if you mixed.
It does the fermentation pathway. It pulls off this carbon and converts the pyruvate to acetaldehyde.
But are we gonna be testing them?
No. No, yes.
Okay.
So you took those because I was running out of time.
The lecture is going to say the pyruvate carboxylase complex is composed of these 3 primary proteins with 3 different functions. Is that also a reason for the lecture?
It's probably accurate in the sense that it's a complex, so there's more than 1 part to it.
Okay.
I'm not sure that it's 3.
Okay.
Yeah, I just know that's not the right end, but,
Okay.
well, that's not the
Should I insert pyruvate dehydrogenase in?
Yes, and then you should look it up and see if it's really made out of 3 parts, 'cause I don't
Okay.
know.
So on your lecture notes, is it the dehydrogenase of pyruvic acid that formed the acetyl-CoA? Is that what it is?
No, it's, okay, so you start with pyruvate, which is a 3-carbon compound. You pull off a carboxyl group, which becomes CO2 and floats away because of diffusion. The 2 remaining carbons get bound to coenzyme A to form acetyl-CoA. And the enzyme that does all of those things is the pyruvate dehydrogenase complex. I know it's a complex, I just don't know how many parts it does.
It's complex.
Yeah. Oh, and the other thing it does is reduce the coenzyme, NAD to NADH. Yeah, make that correction. I should go back and make that correction. Well, okay, so after this, and the significance of this is that acetyl coenzyme A is a high energy compound. Those coenzymes, remember I said coenzyme A? I said acetyl coenzyme A is a high energy compound, succinyl CoA is a high energy compound. The acetyl coenzyme A is going to provide the energy required to stick the 2-carbon group from here to this 4-carbon acid called oxaloacetic acid to make citric acid. Woo-hoo. When that happens, citric acid is made and the coenzyme goes off and it gets to be used again. So now we've entered into the Krebs cycle. Only you don't know it yet. We won't go there. It's time to stop. Knock it off. Citric acid cycle, tricarboxylic acid cycle next time. You guys are doing some good question answering or question asking tonight. That's good. Yippee. Slows me down.
Yay, I'll do it.
Because this is not It's not really straightforward stuff, it's very nebulous, you can't pin it down, you can't look at it with a microscope.